This is the instruction video for Precision Decision Study Group 1, Sites that will centrifuge samples.
This is the Precision Decision Quick Reference Card. As the name suggests, the Quick Reference Card is intended to serve as an easy-to-follow, quick information and instruction guide for staff involved in all aspects of precision decision study at your site. More detailed information and instructions can be found in the MOOC for Precision Decisions, so please refer to the MOOC as needed. Each time a shipment of kits containing blood tubes is sent to your site, you will receive an email notification from the shipping site. Included in that email notification will be a copy of the Quick Reference Card. You can print the two-page document, either single or double-sided, and fold it as shown here if desired. The Quick Reference Card contains information and instructions for research coordinators, phlebotomy, laboratory staff, shipping, as well as contact information. If desired, you can cut your Quick Reference Card and distribute the information to the appropriate staff members at your site.
This is the kit you will use each time a patient is enrolled in Precision Decision. Please see the shipment packing list provided, listing kit contents. You will find the following tubes in each kit, and blood samples will be collected in the following order: P100 first, SST second, CPT third, and Tempus fourth. This is an example of the requisition form and barcode labels you will use for each patient visit. You will print the requisition forms as needed from the provided PDF file. The barcode labels will be supplied by the Toronto site. Please note that 2 identical barcode labels will be provided for each blood tube per patient visit. Barcodes ending with a P will be used for P100 tubes, S for SST tubes, C for CPT tubes, and T for Tempus tubes. Place one barcode label on the appropriate empty tube. And the other identical label on the designated box on the requisition form. Ensure that all tubes receive the correct barcode label and labels are placed correctly on the requisition form. You must record your site's 3-digit CARA site ID on the requisition form at the top left, as well as on the barcode labels associated with the patient visit. The 3-digit CARA site ID can be recorded either before or after the sample has been collected. However, it may be easier to record this information on the requisition and labels before sample collection. Each patient will have white baseline visit labels and corresponding yellow 6-month visit labels. Please ensure that each patient is assigned one set of matching labels: white labels with a 5-digit number ending in 1 for baseline, and matching yellow labels with the same first 4 digits followed by the number 2 for 6-month visits. After blood samples have been collected into the labeled blood tubes, immediately shake each tube as follows and record the blood collection time on the requisition form. For P100 tubes, invert 10 times. For SST tubes, invert 5 times, then let the tubes sit upright at room temperature for 30 minutes to allow blood clotting. For CPT tubes, invert 10 times. For Tempus tubes, shake vigorously for 30 seconds, or if you have a vortex available, vortex for 30 seconds. Remember to record the time of blood collection on the requisition form. Centrifuge SST blood tubes at room temperature for 10 minutes at 1300 G with the brakes on. Record the spin start time on the requisition form. For CPT tubes, centrifuge at room temperature for 20 minutes at 1800 G with the brakes on. Record the spin start time on the requisition form. After centrifugation, SST tubes will look like this, with red blood cells at the bottom, the gel layer in the middle, and serum layer on top. The CPT tube will also have the red blood cells on the bottom, followed by the density solution layer, the gel layer, mononuclear cell and platelet layer, and plasma layer on top. To ship blood samples, place all labeled blood tubes in the tube holder material provided. Wrap them securely in bubble wrap. Place the wrapped samples in the plastic sealable bag provided. Peel off the protective paper and fold to seal the bag such that the star is inside of the square box. Next, place the sealed blood samples in the small cardboard box. Close the box as shown. Then place both the white UN 3373 and orange biohazard stickers on the box. Fill out the FedEx form with the appropriate shipping details and record the FedEx tracking number on the requisition form. Fold and place the completed requisition form in the box containing the sealed blood samples. Place the folded form in the side slot of the sealed blood tubes bag. Send an email notification to the appropriate receiving site to inform them about the sample you will ship. Also attach a copy of the completed requisition form in the email. U.S. sites will ship to Cincinnati and send an email notification to both Rheumatology Tissue Core and CARA email addresses listed here. Canadian sites will ship to Toronto and will send an email notification to the CARA email listed here only. This concludes the Precision Decision instruction video. Thank you for watching.